Hi and welcome to my OCR A-Level Biology revision session with me, Christine. So today's lesson I want to look at cloning in plants. So when we talk about natural cloning, we call it vegetative propagation. Now, what we're going to get is this structure forming, and that's going to be able to develop into a fully differentiated new plant. And this differentiated plant is genetically identical to the parent plant. The reason being it has come through the process of mitosis, so asexual reproduction. And there's a few examples we have of this happening in plants. So we can look at, for example, what's known as stem tubers, your potatoes that you have and we eat. Now, these are what we call stem tubers. They are underground swollen stem tips. Now, it's important to note this swollen stem tip is going to be a storage organ, and that storage organ is going to store lots of starch. So the stem tubers are a storage organ, and that then can produce these buds. We call them the eyes of the potato, and the eye of the potato will then be able to start to divide by mitosis to produce more genetically identical newly potato plants. So it's really important that we understand that plants have got undifferentiated stem cells that have the capacity to go through mitotic division and then differentiate. We can also see this with bulbs like garlic. So the garlic is actually where the leaf base has swollen. Now again, we've got storage of starch. What you get with the bulbs is that they have the buds internally, and they will wait until conditions are correct before they will start to develop into shoots or roots, depending on what it is that needs to grow for the plant. So it's really important that we understand that the fact that this is a storage organ, this storage organ is going to provide the polysaccharide, going back to our biological molecules topic area, our polysaccharide, which is made up of amylose and amylopectin. Now these can be hydrolyzed down to provide the necessary glucose molecule for respiration, which will allow for the growth to produce these identical, genetically identical, because they have the exact same DNA as the parent plant. We can also look at the likes of what's known as runners. Now these are lateral stems. These are going to grow from the parent plant, and the minute that the stem touches the ground, what will happen is that will then develop some roots, and the roots will start to grow, and that will then cut off the runner, and that will ensure that that then becomes an individual plant on its own. So you can see that with regards to what's known as runners in our strawberries or in our spider plants. So we call them runners because these are these lateral stems that are going to grow, and when they touch the ground, that is where you are able to then see that development of the roots. Now, as gardeners or as horticultural development companies, what we want to do is we want to get mass growth of these. So what we can do is we can cut off our spider plants, we can remove them from these lateral stems or our strawberry plants, and we can then pot them into our plant pots, and that will result in us ending up with natural clones, and we can have lots of them. So that's one way in which we can use this vegetative propagation, natural cloning, to produce lots of food sources to feed the growing population. But obviously the population's growing way too fast, and we need to find alternative ways to feed the general population. And so therefore what we've realized is we can actually do what's known as artificial cloning, and there's two forms that you need to know. The first one is from taking a cutting. Now I've used this rose. I really like roses. I like to look at them. So therefore what I might want to do is I might want to take a cutting of my rose plant. As a gardener, I want to make a genetically identical copy another plant. So what do I need to do? Well, actually, I don't want to cut my flowering stem. I want to ensure that I take a non-flowering stem and I'm going to cut that at a slant. Now, the reason I want the non-flowering part of the stem is I want to encourage the growth of roots. So to encourage the growth of roots, what I need is one that hasn't yet started to flower. So this non-flowering stem is going to be cut at a slant. Now it's important to know I want to do this between the nodes. So I always want to cut at a slant between the nodes, and that therefore means that there is a bud that will develop and produce my genetically identical offspring plant. So what I can do is I can dip this cut section into some rooting powder. Now the rooting powder contains auxin, so this takes us back to our plant hormones and plant responses, in the fact that this auxin is going to generate and encourage cell mitosis, cell division, and then the cell growth, so cell elongation. So by dipping it into the rooting powder, we're giving it hormones to stimulate that mitotic division and that differentiation. Now, what we want to do when we take a cutting is we want to reduce the leaves to about 2 or 4, and the reason being is we want to reduce transpiration. Too much transpiration will be a problem because the plant will not have enough water available. So you want to reduce that transpiration by removing the leaves. Therefore, you are limiting the concentration of stomata. You're going to place it in the soil and you're going to add water to it. Now, it's important that the water that we're adding is not too much water. We don't want to saturate the soil. We need to ensure that it is a controlled amount of water. And what we also want to do is potentially cover our cutting with a bag. Now, this will increase the humidity inside the bag and therefore again reduce transpiration. So if they're going to ask a question about this, it's going to be either expecting you to describe how you could do a cutting to produce an artificial clone, or they'll give you information and expect you to improve on what they have given you. So the other way in which we can do artificial cloning is called micropropagation. So micro, small, propagation, producing large numbers. And these plants are going to be genetically identical from a single parent plant. And we do that through using what's known as tissue culture techniques. So the first thing we have to understand is that we have meristem tissue, which we are going to remove. Now, this can be removed from the shoot tip and it can also be removed from the axial buds. So what we're going to do is we're going to remove these meristematic tissue, this cells that are undifferentiated and have the capacity to go through mitosis and differentiate. So we're going to remove these cells, and what they are now called once they've been removed is explant. Now, explant needs to be sterilized. We need to avoid any contamination, and so to avoid any contamination you need to have sterilized the explants that have been removed and then place them in a sterile culture medium. Now, we tend to use things like agar plates, or we could use a liquid broth. It just depends what it is that you are growing, but it's important you note your keywords: the explant, sterilized, and then placed into the sterilized culture medium. Now again, this culture medium actually contains auxins and cytokinins. Now these are to stimulate again mitosis, because what we want to get is what's known as a callus. Now a callus is a mass of identical cells that is produced inside the plant. So a plant producing a mass of identical cells is called a callus, and what we would then do is we would take that callus and we would split that callus up, divide it to produce tiny genetically identical plantlets. We would encourage them to grow, and once we have got them into the stage where they are plantlets, where they've started to grow shoots and roots, we would then take those tiny plantlets and we would then pot them in compost to grow into small plants, and then we would then plant them out in the fields, or it would therefore be left in the garden centre for people to come and buy. So it's a way in which we can take a very small take a very small sample of the plant, the meristem tissue, and what we are then going to do is we're going to make large numbers of it by propagating it, large numbers, and they are genetically identical. So it's important that you know if they're going to ask you a question about this, it will be looking for you to state your keywords, but also it will be looking for you to discuss arguments for and against this artificial cloning. So you need to think about some advantages. Now, obviously, the most important one is rapid production of large number of individuals. Now, if we're looking at this as a food source, that's really important because we can feed lots more people if we are doing micropropagation. We can obviously propagate individuals with desirable traits. So I said I would take a cutting of that rose plant that I really liked. Well, it means that we can take desirable traits of plants and we can then propagate so that we're making genetically identical offspring from that original plant that has those desirable traits. We can increase the number of rare species. We can produce large numbers of selectively bred individuals and also ones that we may have genetically modified. So again, it's important to note that they will interlink the topic areas when they're asking questions about this. We can produce seedless plants. They would be a way of propagating a sterile plant, a plant that cannot produce fertile offspring. So therefore it's seedless. We are going to still be able to propagate it. And obviously growing infertile plants like orchids. So orchids actually are infertile and yet we can grow them because we can use micropropagation. We can increase the number of rare or endangered plants as long as we have a sample. And it's quicker. It's much quicker than growing from the seeds because remember, going back to your hormonal topic area, the seeds will lie dormant until there is the right conditions and then they will be activated to produce gibberellins. Gibberellins will then cause the production of proteins. The proteins, the enzymes, will then be needed to break the starch molecule down into the monomers to glucose to allow for the growth. So by doing this, by doing it through the cloning, artificial cloning, we are doing this as a quicker way because it's already got the cells ready to go. It is just needing to produce more cells and then differentiate into the likes of roots if we're taking a cutting, or if we're talking about the explant then going through this division to produce lots of it. So it's important to note that we don't need to break anything down to then allow for the growth, which makes it a quicker process. Now, it's important as well to note whenever you're asked a question on this, that it's not good enough to just say it's quicker than growing from the seeds. It's important you can give a way in which it's quicker whenever they're looking for you to discuss those arguments for and against. Also, it'll allow for the growth of pathogen-free individuals because remember, we were sterilizing it. We sterilized the explant and we also put it into sterile conditions in the culture medium. So it's important to note. Now, disadvantage, obviously they are clones. They are genetically identical. Therefore, a disadvantage is there is a lack of genetic variation. They are monocultures. So what does that mean? Well, that means that the population's at a greater risk of environmental change. If there is a pathogen that comes along, if that pathogen wipes out one of the plants, if they are genetically identical, it will wipe out all of the plants. And that's where you have, for example, the potato late blight protoctistin could come along and wipe them all out if they are genetically identical. Again, how they could interlink those topic areas together. Now, the explants, we're obviously going to be scraping them off of the parent plant. They are vulnerable to infection, hence the need for the sterilized conditions. If the parent plant was actually infected with microorganisms, then one of the disadvantages of artificial cloning is that the offspring plants will also be infected with those potential microorganisms. And obviously we talked about a way to reduce that by doing this sterilized process, the aseptic procedure, but that is complex, which means it's expensive and requires skilled workers. So again, it's really important whenever you are trying to describe a disadvantage that you don't just say it's expensive, that you go on and give a reason for that answer. It's expensive because you need more equipment, and also you require skilled workers that you will have to have trained. So always make sure that you are giving more than just stating expensive, quicker, etc. So I hope you've liked this video, and if you have, then please do click on the like button and subscribe to my channel. And if you haven't already done so, please do check out my revision platform, www.aiqchat.com.